In Chapter 4, we are going to talk about cell structure, and we are going to compare and contrast the different elements of animal and plant cells. Cells are the basic building blocks for all organisms, and your body is made up of many different types of cells, and each cell is specialized for a specific purpose. You can see some examples of some different types of cells. Figure A is a view from a light microscope of some nasal sinus cells. Figure B is actually some onion cells, again viewed with a light microscope, different type of stain here. And then figure C is a specific type of bacterial cell that's actually seen through a scanning electron microscope, and we're going to look at some other scanning electron microscope pictures as we go through this lecture. Organisms are classified based upon the cell types and the cell numbers and the cell constituents that make them up. So us as humans, we are considered multicellular organisms, as we have many different types of cells that have many different specialties. Again, cells are the basic unit of a living thing. So again, humans, we have many cells, or there are also organisms that exist that have only a single cell, like a bacterial organism. Regardless, whether or not an organism has multiple cells or a single cell, if the organism's most simplistic unit is the cell, it is considered a living organism. And we've talked about why viruses are not considered living organisms. So there are several cells of one kind that interconnect with each other and perform a shared function, and these form tissues. So a bunch of cells make up tissues. Then these tissues combine to form an organ. Organs such as our stomach, our pancreas, our spleen, heart, liver, lungs, kidney, brain, things like that. And then the organs work together to make up organ systems. We've talked about the cardiopulmonary system that is comprised of the heart and the lungs. And then all of these systems together, they function and maintain homeostatic balance, hopefully, to form the entire organism. As you may know already, cells can vary in their sizes and their shapes. And with a few exceptions, we cannot see individual cells with our naked eye. So scientists use microscopes to study cells, micro meaning small and scope meaning to look at. A microscope is an instrument that magnifies an object, and when we take pictures or photographs with microscopes, we make images known as micrographs. And in this picture here, you can see two different types of microscopes. The first one you all may be very familiar with in Figure A. This is a common light microscope that you can find in a college or university biology lab. And this can magnify cells approximately up to 400 times, and they usually have a resolution at about around 200 nanometers. This more complex instrument over here on the right-hand side, I actually had the privilege to work with one of these for my graduate work. This is an electron microscope, and this provides a much higher magnification. This can magnify objects up to 100,000 times and has a resolution of 50 picometers. On this slide, I just want to talk about some really important definitions that you all should be familiar with in terms of magnification and resolving power when we talk about microscopy. So magnification is the process of enlarging an object in appearance. And resolving power is the microscope's ability to distinguish two adjacent structures as separate. So the higher the resolution, the better the image's clarity and detail. So you can see and compare here this radiolarian under a light microscope versus the radiolarian under the electron microscope. You can clearly see the differences in these cavities right here and the differences in the borders between the different aspects of this organism right here. So much clearer picture in the electron microscope compared to the light microscope. So we can say that the electron microscope has better resolution than the light microscope does. In contrast to light microscopy, electron microscopes can achieve a higher magnification and a better resolution because instead of using a beam of light, they use a beam of electrons to create the image. So there are many different types of methods that are used to prepare cells for electron microscopy, and these are very detailed and very sensitive protocols, and it's kind of beyond the scope of this class, but if you're interested, you can do a Google search of some protocols for how to prepare different cell types for electron microscopy. And what we're visualizing here, the figure that I'm drawing this little star above, this is actually a transmission electron microscope image of a mitochondria. So you can see the different cristae in the mitochondria here. And then the scanning electron microscope helps us to visualize the three-dimensional exterior views of different types of cells. So in this picture down here, you can see the biconcave disc shape of a red blood cell or a couple of red blood cells that are visualized using scanning electron microscopes. So now let's move on to the cell theory. And this is an extremely important underlying principle of biology, and you will see questions about the cell theory on your upcoming quizzes and exams. So the cell theory came about through many, many years of research and data and articles published by many different scientists to come up with these three basic principles of what cells are and their basic overall importance. So first of all, cells are the basic unit of life, number one. All living organisms must be made of cells. This is why a virus is not considered alive, because viruses are technically not made of cells. And then all cells must come from pre-existing cells. Cells don't just appear out of thin air. They have to come from some type of pre-existing cell. It's also very important for you all to understand that all cells share four common components. The first being a cell membrane. Your textbook calls this a plasma membrane, and this membrane helps to separate the cell's interior from the exterior, helps to keep the cell's interior components from mixing with the exterior or the extracellular environment. All cells have a cytoplasm, which is made of cytosol. All cells have DNA, and we're going to talk about where DNA can be found in prokaryotes versus eukaryotes. And all cells also have ribosomes, which are responsible for protein synthesis. Now let's talk about some of the characteristics of prokaryotic cells. So it's very important for you all to understand the differences between prokaryotic cells and prokaryotic organisms such as bacteria and eukaryotic organisms such as us humans. So first off, prokaryotes, they lack membrane enclosed internal compartments, so they don't have a true nucleus. So you may be asking the question, well, isn't DNA found in the nucleus in eukaryotes? Yes, it is. But DNA is present in prokaryotes, it's just found in a different location. It's actually found in a structure known as the nucleoid. And most prokaryotics have a peptidoglycan cell wall, and many have a polysaccharide capsule. So this cell wall is really important for protection, and it helps the cell to maintain its shape, and it also prevents against dehydration. Some prokaryotes have other components like flagellum and pili and fimbriae. The flagella are used for locomotion. The pili exchange genetic material during conjugation, and the fimbriae are used to attach to a host cell. And when we talk about the classification of organisms, when we talk about what domain and what kingdom and so on, how to classify the nomenclature of organisms, we're going to be talking about the domains archaea and bacteria for prokaryotic organisms. And because prokaryotic organisms are so simplistic, it is hypothesized and theorized by many scientists and many textbooks that these may have very well been the first types of organisms that existed on planet Earth. Another important component of a characteristic for prokaryotic prokaryotic organisms is that their ribosomes are found in the cytoplasm, and they have their cell membrane surrounded by a cell wall. Prokaryotic cells are much smaller than eukaryotic cells, and this small cell size allows for ions and organic molecules that enter them to quickly diffuse to other parts of the cell. Also, any waste products that are produced within a prokaryotic cell can quickly diffuse. This is not really the case with eukaryotic cells, which have developed different structural adaptations to enhance intercellular transport. So small size in general is very important and necessary for all types of cells, whether they are prokaryotic or eukaryotic. So why is that? Well, first let's talk about the area and the volume of a typical cell. Not all cells are spherical in shape, but most tend to look like a sphere or approximate a sphere. And you may recall from high school geometry that the formula for the surface area of the sphere is 4 pi times the radius squared, while the formula for its volume is 4 pi times the radius cubed all over 3. So as the radius of a cell increases, its surface area increases as the square of its radius, but its volume increases as the cube of its radius. So the volume increases much more rapidly. So as a cell increases in size, its surface area to volume ratio decreases. And the same principle would apply if the cell had a cuboidal shape. So if the cell grows too large, then the plasma membrane will not have sufficient surface area to support the rate of diffusion that's required for the increased volume. So as a cell grows, it actually becomes less efficient. And one way for a cell to become more efficient is to divide. We all know cells are capable of growth, and it is inevitable for cells to grow, so they have to divide. Other ways are to increase their surface area by foldings of the cell membrane. They can flatten themselves, they can become thin or elongated, or develop specialized organelles that perform specific tasks. And all of these adaptations combined lead to the development of more sophisticated cell types, which we call eukaryotic cells. Now that we have talked about the small, simplistic prokaryotic cells, we're going to move into the more highly organized eukaryotic cells that make up humans and other complex organisms. So importantly, eukaryotic cells have a membrane-bound nucleus where our DNA can be found. There are many different types of membrane-bound organelles, such as the endoplasmic reticulum, the Golgi, the mitochondria, so on and so forth. And there are rod-shaped chromosomes. Because a membrane surrounds the eukaryotic cell's nucleus, we can say that eukaryotic cells have a true nucleus. And you're going to hear me say the word organelle constantly throughout this talk and throughout the semester. Organelle simply means little organ. And as I mentioned previously, organelles have specialized cellular functions, just as your body's organs have specialized functions. So the organelles work together in the cells to regulate the cell and maintain homeostasis, and they contribute to the global organ function eventually. So don't worry about getting all the tiny components that are labeled here on this picture. We're going to go through the important ones in the upcoming slides. And just like prokaryotes, eukaryotes also have a plasma membrane. And you can see this plasma membrane, this phospholipid bilayer that's embedded with proteins to help separate the internal contents of the cell from its surrounding environment. A phospholipid is a lipid molecule with 2 fatty acid chains and a phosphate-containing group. The plasma membrane controls the passage of organic molecules, ions, water, and oxygen into and out of the cell. And also wastes such as carbon dioxide and ammonia. They leave the cell by passing through the plasma membrane. The cytoplasm is the cell's entire region between the plasma membrane and the nuclear envelope. It's comprised of organelles suspended in the gel-like cytosol. the cytoskeleton, and various chemicals. Even though the cytoplasm is made up of 70 to 80% of water, it has a semi-solid consistency which comes from the proteins within it. However, the proteins are not the only organic molecules in the cytoplasm. Glucose and other sugars, polysaccharides, amino acids, nucleic acids, fatty acids, and glycerol derivatives can also be found there. Ions of sodium, potassium, calcium, and many other elements also dissolve in the cytoplasm. And there are many metabolic reactions, such as protein synthesis, that take place in the cytoplasm as well. The nucleus is usually the most prominent organelle in a cell, and we can say nuclei for the plural form. So the nucleus houses the cell's DNA in eukaryotic cells and directs the synthesis of ribosomes and proteins. The nucleus stores chromatin, and within the nucleus we can find the nucleolus, which is a condensed chromatin region where ribosome synthesis occurs. And we call the nucleus's boundary the nuclear envelope. It consists of two phospholipid bilayers, an outer and an inner membrane. The nuclear membrane is continuous with the endoplasmic reticulum, and there are nuclear pores that allow substances to enter and exit the nucleus. The nuclear envelope is a double membrane structure that constitutes the nucleus's outermost portion. So both the nuclear envelope's inner and outer membranes are known as phospholipid bilayers. The nuclear envelope is punctuated with pores that control the passage of ions, molecules, and RNA between the nucleoplasm and the cytoplasm. The nucleoplasm is the semi-solid fluid inside the nucleus where we can find the chromatin and the nucleolus. So we've already mentioned that the nucleus directs the synthesis of ribosomes, but how exactly does it do this? Some chromosomes have sections of DNA that encode ribosomal RNA, or rRNA. And specifically, the darkly stained area within the nucleus, also known as the nucleolus or nucleoli in the plural form, they aggregate the ribosomal RNA with associated proteins to assemble ribosomal subunits that are then transported out through the pores in the nuclear envelope to the cytoplasm. Ribosomes are the cellular structures that are responsible for protein synthesis, and they are made up of two different size subunits. So we have the large ribosomal subunit here, and we have the small subunit right here. And ribosomes, as I mentioned previously, they're found in both prokaryotic and eukaryotic organisms, but they are slightly larger in eukaryotic organisms. And they are made up of special RNA, also known as ribosomal RNA or rRNA, and many different types of proteins. And during protein synthesis, the ribosomes assemble amino acids into proteins. Mitochondria, or in the singular form mitochondrion, are known as the powerhouse of both plant and animal cells because they are responsible for making adenosine triphosphate, or ATP. ATP is the cell's main energy-carrying molecule. So mitochondria are oval-shaped. They are double membrane organelles, as you can see here in this electron microscopy image. There's an outer membrane and an inner membrane. And they actually have their own ribosomes and DNA. So they have their own genome, essentially. Each membrane, both the outer and the inner membranes, are made up of a phospholipid bilayer embedded with proteins. The inner layer has folds known as cristae, so I pointed cristae out on the other electron microscopy image. And we call the area surrounded by the folds the mitochondrial matrix. So all this is considered the mitochondrial matrix. And the cristae and the matrix have different roles in cellular respiration. Peroxisomes are small, round organelles enclosed by single membranes, and they carry out oxidation reactions that break down fatty acids and amino acids, and they also may help detoxify many poisons that humans put into their body, such as alcohol. Not that any of you have ever drank alcohol or anything like that before. So at this point, you know that each eukaryotic cell has a plasma membrane, cytoplasm, a nucleus, ribosomes, mitochondria, and peroxisomes. But there are some striking differences between animal and plant cells. While both plant and animal cells have what's known as MTOCs, microtubule organizing centers, animal cells also have centrioles associated with the MTOC, and this makes up a complex that we call the centrosome. Animal cells each have a centrosome and lysosomes, whereas most plant cells do not. Plant cells have a cell wall, chloroplasts, and other specialized plastids, and a large vacuole, where animal cells do not. The centrosome is an MTOC, or a microtubule organizing center, found near the nuclei of animal cells. It contains a pair of centrioles, which are two structures that lie perpendicular to each other. And each centriole is a cylinder of 9 triplets of microtubules. The centrosome replicates itself before a cell divides, and the centrioles appear to have some role in pulling the duplicated chromosomes to the opposite ends of the dividing cell. However, the centriole's exact function in cell division isn't clear because the cells that have had the centrosome removed can still divide. And in plant cells, which lack centrosomes, they are still capable of cellular division. The plant cell wall is a rigid covering that protects the cell. It also provides structural support and gives shape to the cell. Fungal and some protozoan cells also have cell walls. While the prokaryotic cell wall's chief component is peptidoglycan, the major organic molecule in the plant and some protist cell wall is cellulose, which is a polysaccharide composed of glucose units. So similar to mitochondria, chloroplasts also have their own DNA and ribosomes, but they have an entirely different function. The most important function for chloroplasts is to carry out photosynthesis. And this is the series of reactions that use carbon dioxide, water, and light energy to make glucose and oxygen. And this is a major difference between plants and animals. Us as human beings, there's no essential need for us to carry out photosynthesis. Plants are also known as autotrophs, which means they can generate their own food source. Humans are known as heterotrophs. We cannot generate our own food source, so we need to ingest our food for energy. And very similar to mitochondria, chloroplasts have both inner and outer membranes. But within the space enclosed by a chloroplast's inner membrane is a set of interconnected and stacked fluid-filled membrane sacs that we call thylakoids. And you can see the thylakoids here. And each thylakoid stack is a grana, or in plural form, it's grana. We call the fluid enclosed by the inner membrane that surrounds the grana the stroma. And this is the aqueous fluid, the stroma, all in here. The central vacuole plays a key role in regulating the cell's concentration of water in changing environmental conditions. If you ever notice that if you forget to water a plant for a few days, it will start to wilt, that's because as the water concentration in the soil becomes lower than the water concentration in the plant, water moves out of the central vacuoles in cytoplasm. And as the central vacuole shrinks, it leaves the cell wall unsupported. And this loss of support to the plant cell walls results in the wilted appearance. The central vacuole also supports the cell's expansion. When the central vacuole holds more water, the cell becomes larger without having to invest considerable energy in synthesizing new cytoplasm. The endomembrane system is a group of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, the lysosomes and vesicles, and the endoplasmic reticulum and Golgi apparatus. Although not technically within the cell, the plasma membrane is included in the endomembrane system because it interacts with the other endomembranous organelles. The endomembrane system does not include either mitochondria or chloroplast membranes. Lysosomes play an important role in the digestive component and the organelle recycling facility of animal cells, and they are considered part of the endomembrane system. They use their hydrolytic enzymes to destroy pathogens that might enter the cell. A good example of this, you can see in the picture here, occurs in macrophages, a group of white blood cells which are part of your body's immune system. So in the process known as phagocytosis or endocytosis, what happens is a section of the macrophage's plasma membrane invaginates or folds in and engulfs a pathogen. The invaginated section with the pathogen inside then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle then fuses with a lysosome, and the lysosome's hydrolytic enzymes help to destroy the pathogen. The endoplasmic reticulum, or the ER, is a series of interconnected membranous sacs and tubules that collectively modify proteins and synthesize lipids. It's important to remember that these two functions take place in separate areas of the endoplasmic reticulum, the rough ER and the smooth ER. So in the rough ER, this is where the ribosomes are going to be found, and this is the area that's going to undergo protein synthesis. And in the smooth ER, there's not going to be ribosomes, so it's going to have a more smooth appearance, and this is the area where lipids are synthesized. We call the endoplasmic reticulum's tubules hollow portion the lumen or the cisternal space. The endoplasmic reticulum's membrane, which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. So the rough endoplasmic reticulum is named as such because the ribosomes attached to its cytoplasmic surface give it a studded appearance when viewing it through an electron microscope. Ribosomes transfer their newly synthesized proteins into the rough endoplasmic reticulum's lumen, where they undergo structural modifications. These modified proteins incorporate into cellular membranes, such as the endoplasmic reticulum or the endoplasmic reticulum's other organelles membranes. The proteins can also secrete from the cell. The rough endoplasmic reticulum also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the rough endoplasmic reticulum, they will reach their destinations via transport vesicles that bud from the rough endoplasmic reticulum's membrane. Since the rough endoplasmic reticulum is engaged in modifying proteins such as enzymes that secrete from the cell, you would be correct in assuming that the rough endoplasmic reticulum is abundant in cells that secrete proteins. This is the case with liver cells, for example. And the smooth endoplasmic reticulum is continuous with the rough endoplasmic reticulum, but it has very few, if any, ribosomes at all on its cytoplasmic surface. And again, this is where lipid synthesis occurs. Also, what other processes that occur here include carbohydrate and steroid hormone synthesis. Also, detoxifications of medications and poisons occur in the SER, and the SER also has the capability to store calcium ions. In muscle cells, a specialized smooth endoplasmic reticulum known as the sarcoplasmic reticulum is responsible for storing calcium ions that are needed to trigger the muscle cell's coordinated contractions. The sorting, tagging, packaging, and distribution of lipids and proteins takes place in the Golgi apparatus. The side of the Golgi apparatus that is closer to the endoplasmic reticulum, I'm going to highlight this one right here, so this side right there, is called the cis face. The opposite side, that's out here, is called the trans face. The transport vesicles that formed from the endoplasmic reticulum travel to the cis face, they fuse with it, and then they empty their contents into the Golgi apparatus's lumen. As the proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is adding short sugar molecule chains. These newly modified proteins and lipids then tag with phosphate groups or other small molecules in order to travel to their proper destinations. Finally, the modified and tagged proteins are packaged into secretory vesicles that bud from the Golgi's transphase here. So this is where the budding occurs from the transphase. While some of these vesicles deposit their contents into other cell parts where they'll be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. The network of protein fibers found within the cytoplasm are known as the cytoskeleton. And there are three types of fibers within the cytoskeleton. We have the intermediate filaments, we have the microfilaments, and we have the microtubules. And we're going to talk more about each one of these in depth, but overall the cytoskeleton helps to maintain the shape of the cell. It helps to hold organelles in some specific positions. It allows for the movement of cytoplasm and vesicles within the cell, and it enables cells within multicellular organisms to move. Of the three types of protein fibers that make up the cytoskeleton, microfilaments are the narrowest. They function in cellular movement and have a diameter of about 7 nanometers, and they're comprised of two globular protein intertwined strands, which we call actin. So for this reason, we can also call microfilaments actin filaments. Microtubules are small hollow tubes. Polymerized dimers of alpha-tubulin and beta-tubulin, which are two globular proteins, comprise the microtubules' walls. Microtubules are the cytoskeleton's widest components. They help the cell resist compression. They provide a track along which vesicles move throughout the cell, and they pull replicated chromosomes to opposite ends of a dividing cell. And like microfilaments, microtubules can disassemble and reform very quickly. They're also the structural elements of flagella, cilia, and centrioles. In animal cells, the centrosome is the MTOC, or the microtubule organizing center. In eukaryotic cells, flagella and cilia are quite different structurally from their counterparts in prokaryotes. The flagella that we can see right here are long hair-like structures that extend from the plasma membrane and enable an entire cell to move. In contrast, cilia are made up of many of these hair-like structures that extend along the entire length of the plasma membrane that move entire cells. But despite the differences in length and number between flagella and cilia, they do share this common structural arrangement of microtubules. They have the same ultrastructure. And the ultrastructure that they share is known as the 9+2 array of microtubules. So there are 9 doublets on the outside, and this is an electron microscopy picture of a sperm cell. So 1, 2, 3, 4, 5, 6, 7, 8, 9 doublets on the outside and 2 unfused in the center. So this is the 9+2 array of microtubules that are found in both cilia and flagella. While cells in most multicellular organisms release materials into the extracellular space, we're going to discuss animal cells as an example here. The primary components of these materials are proteins, and the most abundant protein is collagen. Collagen fibers are interwoven with proteoglycans, which are carbohydrate-containing protein molecules. Collectively, we call these materials the extracellular matrix. Not only does the extracellular matrix hold the cells together to form a tissue, but it also allows the cells within the tissue to communicate with each other. Cells can also communicate with each other through direct contact or intercellular junctions. There are differences in the ways that plant and animal and fungal cells communicate. The Plasmodesmata are junctions between plant cells, whereas animal cell contacts include tight junctions, gap junctions, and desmosomes, which we're going to talk about on the upcoming slides. Plasmodesmata are channels that pass between cell walls and plants to connect cytoplasm and allow materials to move from cell to cell. A tight junction is a watertight seal between two adjacent animal cells. Proteins tightly hold the cells against each other. This tight adherence prevents materials from leaking between the cells, and tight junctions are typically found in epithelial tissues that line internal organs and cavities and comprise most of the skin. For example, the tight junctions of the epithelial cells lining your urinary bladder prevent urine from leaking out into the extracellular space. Desmosomes are exclusively found in animal cells, and these act like spot welds between adjacent epithelial cells. Cadherins are short proteins in the plasma membrane that connect to intermediate filaments to create desmosomes. The cadherins connect two adjacent cells and maintain the cells in a sheet-like formation in organs and tissues that stretch like the skin, heart, and muscles. Gap junctions in animal cells are similar to the plasmodesmata that are found in plant cells because they are the channels between adjacent cells that allow for the transport of ions, nutrients, and other substances that enable the cells to communicate. However, structurally, there are some differences between the gap junctions and plasmodesmata. The gap junctions develop when a set of 6 proteins, known as connexins, in the plasma membrane arrange themselves in an elongated donut-like configuration, also known as a connexon. When the connexon's pores, or these donut holes in adjacent animal cells, align, a channel between the two cells forms. Gap junctions are particularly important in cardiac muscle. The electrical signal for the muscle to contract passes efficiently through gap junctions, allowing the heart muscle cells to contract in tandem.